Hey guys, this is blood vessel anatomy video number 4, our last one to look at blood vessel anatomy. So let's get back to our PowerPoint. And here's where we left off. So we had just looked at all the different types of blood vessels, but now we're just going to spend a little bit more time talking about capillaries and capillary beds, because capillary beds are actually pretty interesting areas inside the body. So here's something we haven't mentioned yet, that the capillaries have these structures called precapillary sphincters. Sphincters are little muscular circles inside the body that can constrict and stop flow. So you have a sphincter in your urethra and you have sphincters in the anus. Because obviously, do you want to walk around the world peeing and pooping constantly? No. You want the sphincter to be closed. So here's the tube. The sphincter is kind of like the valve that keeps the tube closed. And then when you're in an appropriate environment, you can relax the sphincter, and then whatever's in the tube can come through, right? So this is like the process by which we can control urine output from the body and fecal output from the body. So we have sphincters in the anus and the urethra. But we also have sphincters in these capillaries. So why would a sphincter be useful in this case? So here's the blood coming through the arteriole, and it's coming into this capillary bed. Now, when the sphincters are open, these little channels are open, the blood can flow through all the different channels of the capillary bed. So see how it's coming up here, going through this area, coming through here, going down here. So the blood is flowing through all the tiny channels in the capillary bed when the sphincters are open. So that means that this tissue right here is getting lots of oxygen, because as the blood is coming through, it's delivering the oxygen to the surrounding tissues. So if the sphincters are open, the tissue's getting oxygen, and it's probably a hard working tissue, because tissues that work hard are the ones that need the oxygen. Now, what if my tissue was resting? What if this tissue right now has got plenty of oxygen, not much CO2, plenty of nutrients? Do we need to send the blood to this capillary bed if the tissue already has everything it needs? No. So in this case, you close the sphincters. See, it says sphincter closed. You close this sphincter, you constrict that little muscle, and now the blood is not going through all of this. It's going straight through the middle and out the other side, right? So the channel, it's going right through the middle, and now we could potentially send this blood somewhere else rather than going to this particular area, to this resting tissue. So usually in resting tissues, the sphincters are closed because this tissue doesn't need anything. It has oxygen, it doesn't have waste products, it's got nutrients, so it doesn't need any more blood coming through here. It's already got everything it needs. But if it switches from resting to working, all you have to do is open the sphincters, and now the blood will be delivered to all the areas inside the tissue. So let's see how this could be useful in the body. So here is my resting tissue. Here is my hard working tissue, right? Do I want to send blood to the resting guy, or should I send it to the hard working guy? I should send it to the hard working guy. He needs it. So he needs oxygen. He needs nutrients. And he has to get rid of waste products. So basically, he needs a blood supply coming through to deliver oxygen and nutrients and pick up the waste products. So in this case, what am I going to do in my hard-working tissue? I am going to open sphincters. Okay? And I want the blood to flow through all my tissue, and I want all my cells to get oxygen and nutrients. Now this guy, do I want to waste oxygen and nutrients on this resting tissue, or should I keep it going down here until it gets to the working tissue? I would prefer the working tissue get the nutrients rather than this guy who doesn't need them. So again, you're going to use your distributing arteries here to switch up the flow, and then the sphincters will also help you by preventing this guy from getting the oxygen and nutrients when he doesn't need them. We want to keep sending them down towards the hard working tissue. So hopefully you get the concept and why those sphincters are helpful. So let's look at the different types of capillaries. There's 3 different types: continuous, fenestrated, and sinusoids. So continuous means that there is no space between cells. Fenestrated, fenestra, means window. So there are small windows in the cells and between the cells, both. And then sinus is a large space. So let's look at the pictures. So here's continuous. So here's my red blood cell in the continuous capillary. There is no way for anything to get from in here to outside. These aren't spaces. They're tight junctions. Remember we learned about tight junctions in biology 203? Could draw a little bit up here. Here's one cell. Here's another. If you have tight junctions, they're tightly glued together. So can anything go between the cells to get to the other side? No. The only way something can get through is to pass through the cell and come out the other side. And this is really valuable because as it passes through, we can check it. So we can make sure that we want to send this thing out or in. It can go either way. But the message is it has to go through the cell, not between the cells, because there are tight junctions. So these type of junctions were good in intestines and places where you don't want things moving in and out of blood vessels. And now let's look at our fenestrated. Remember, fenestra is a window. So look at these holes. See how there's holes everywhere in my cell? These are the fenestrations or the pores. So see it says fenestrations or pores. You could just call them plain old holes, right? They're small little holes, small windows. So now if something is in here, can it go outside through the holes? Yes, it can if it's small enough, right? So if it'll fit through this hole, it can go in to the blood vessel. And if it'll fit through this hole, it could come out of the blood vessel. So small things can go in and out. Can my blood cell go through this hole? It's much too big, right? So small things like nutrients are going to go through these little holes. So that's a fenestra, a fenestrated capillary. They're important in the kidney. We're going to see them, talk about them a lot in the kidney. And now here's our third type, the sinusoidal capillary. Sinus is a big hole. So see there's a giant hole here. So these are found in the liver, in bone marrow, and in the spleen. Capillaries with giant holes in them. So things can very easily go in and things can very easily come out, right? Through these giant holes. So we're going to see why those are important when we study how the liver works and when we study how the spleen works. So continuous, we have the tight junctions. The cells are tightly glued together, like we learned in biology 203. There's very tight connections here. They're stuck together. So if something wants to go from over here to over here, how does it get through? It has to pass through the cell. It can't go between the cells, right? So this is our continuous capillary. Our fenestrated capillary has tiny windows, tiny holes in it. So anything that's small enough to go through the hole could potentially come out of the blood vessel or come out of the capillary. So here's our continuous one with the tight junctions between the cells, right? Here's our fenestrated one with the little holes all over the cells. See the little pores everywhere? So anything that's small enough to go through the hole is going to be able to come out of the capillary. And remember, capillaries are just large enough to fit one red blood cell inside. Can the red blood cells come through the fenestrae? Nope, too big. And the fenestrae are only for small, tiny little molecules. This is showing you what the capillary looks like under the microscope. Here's a little window, here's a little window, Here's a little window. There are actual pores or tiny windows in the wall of the endothelial cell. So here's the sinusoids. Remember, they have giant holes, so they're very leaky capillaries. They have really large, the sinusoids are very leaky. So they have large holes in them. Okay, so basically anything can go in and out of these guys because the holes in the spaces are so huge. So they're really important in the liver, in the bone marrow, and in the spleen. And in the liver and the spleen, the macrophages are lining the sinusoids. So what's a macrophage? It's one of those large cells from the immune system. It can act like a Pac-Man. If it sees something dangerous, like a bacteria, it can eat and destroy it. So phage means to eat, macro means large. So it's a large cell that eats things, a macrophage. So usually inside the liver and inside the spleen, the sinusoids, these capillaries are lined by macrophages. And you see, we would want, if there's something nasty in this blood coming through the liver, we would want it to leak out here so the macrophage could see it and eat it. We want it to be noticed. So that's why we need these holy capillaries, these capillaries with the giant holes in them. So here's what it looks like in the liver. Blood is coming from the digestive system into the hepatic portal vein, and now it has to pass through the liver tissue to get into the hepatic venule, hepatic vein, and then it can come back to the heart. What's the function of the liver? It's supposed to clean up, right? It's supposed to get rid of toxins, get rid of dangerous things. Are we sure that everything we absorbed in the digestive system was good? So this blood is coming from the digestive system. Are we sure that we can give this the gold star, that everything is clean and perfect, that there's no bacteria, no viruses, nothing weird or strange that we absorbed in our digestive system? No, we can't do this, right? We can't give this the seal of approval. We could have absorbed anything in the digestive system. Some weird and wonderful things could have come in with that food. So do we want to send blood from the digestive system straight to the heart? If there's bacteria and viruses, do we want those going into the heart? Definitely not. So you want the blood from the digestive system to first go to the liver through this hepatic portal vein, right? And then it passes through the liver tissue. What did I tell you was lining all of these sinusoids or all of these large capillaries with the spaces? So all along here, I have my macrophages, my cells that are waiting to eat the strange and weird and wonderful things. So the macrophages are going to eat anything strange, toxic, dangerous, get rid of it. So now the blood coming into the hepatic venule is nice and clean. And now we can send it through the hepatic vein into the inferior vena cava and into the heart. So that's why we have this hepatic portal circulation. We're not sure that the blood coming in from the digestive system is clean, so we're going to send it through the liver to be checked and cleaned. It's going to come out the other side. Hopefully it'll be clean by now, go through the hepatic vein, and then back to the heart. So we see these sinusoids or capillaries with large holes in them inside the liver. Okay, so that's everything about blood vessel anatomy from our slides. So we'll save a lot of lecture time by doing these right now and not in class. And if you have any questions, of course, we can go over some aspects of this again in class, but hopefully I explained it clear enough that you understand the major gist of it, and we're going to do some worksheets in class to make sure that everybody gets the key points. So see you in class soon. Have a nice day.